It sounds like something straight out of science fiction, doesn't it? The idea that the very first spark of life can happen not in the body but inside a laboratory dish? Well, that's the incredible reality of in vitro fertilization, or IVF. Today, we're going to dive into the amazing journey of biology and technology that makes it all happen. So, the name itself, in vitro fertilization, kind of gives the game away. In vitro is just Latin for in glass. It's such a simple phrase for an idea that is just profound. Life beginning in the controlled, precise environment of a lab. Now, this didn't just happen overnight. The groundwork was actually laid with animals. Yep, the first IVF offspring were rabbits, way back in 1959. But the game changer for humans? That was 1978, with the birth of Louise Brown. All of a sudden, what seemed impossible was real. And decades later, the pioneer who made it happen, Dr. Robert G. Edwards, was deservedly awarded the Nobel Prize. Okay, so that brings us to the big question, right? How in the world do scientists and doctors actually orchestrate the very beginning of a new life inside a lab? Well, let's break it down, step by step. The whole thing really starts long before anyone even sets foot in the lab. The first major step is all about preparation, getting the body ready to give the best possible chance for success. And this stage, it is absolutely crucial. It really boils down to a two-part mission. First up is superovulation. That's where hormones are used to encourage the ovaries to produce not just one egg, like in a normal cycle, but a whole bunch of them. And then comes the second part, the egg retrieval, which is the procedure to carefully collect all those eggs. Superovulation is really all about playing the odds. Instead of just one egg, the goal is to get many. So for about 10 days, a series of hormone injections are used to stimulate the ovaries to grow multiple mature follicles. Each one of those follicles holds a potential egg. It's kind of like a numbers game, and this is step one to stacking the deck. And then the timing becomes everything. A final injection, they call it the trigger shot, kicks off a countdown. It perfectly mimics the body's natural hormone surge to put the finishing touches on the egg's maturation. This opens up a super narrow window of opportunity. The retrieval has to happen, no exceptions, between 34 and 36 hours later. Any earlier, the eggs aren't ready. Any later, the body might release them on its own. The retrieval itself is an incredibly delicate procedure. A doctor uses an ultrasound to guide a needle right to the ovaries to collect the eggs, hoping for a good number, somewhere between 10 and 30. Now, while that's going on, the semen sample is getting its own prep. There's a process called sperm washing that gets rid of inactive cells and isolates only the strongest, most active swimmers, getting them ready for their moment. So, with the eggs and sperm prepped and ready, the action shifts from the clinic to the lab. This is where the magic really happens. This is the moment of creation. Think about these numbers for a second. In a standard IVF setup, each egg is introduced to about 75,000 sperm in a special dish. It's kind of like a huge party, and they're all left together for about 18 hours to see what happens, letting nature take its course, just outside the body. So what are the embryologists looking for? What's the sign that it worked? Well, it's this moment right here. You can see these two little circles inside. Those are the male and female pronuclei. Think of them as the little packages of genetic material from the sperm and the egg. When they fuse together, boom, that's it. Fertilization is confirmed. The second it's fertilized, it gets a new name. It's an embryo. From there, it's moved into a special growth medium and cultured inside an incubator for a couple of days. The whole time, embryologists are watching it like a hawk, waiting for it to divide and grow to that critical 6 to 8 cell stage. And just look at this. This is what it's all for. You're looking at a microscopic image of a human embryo at that 8-cell stage. It is just an incredible and honestly a humbling peek into the very first moments of a potential new life, all happening because of this amazing technology. So, after a few days of very careful watching and growing in the lab, we've arrived at the final clinical step. It's time to transfer the embryo back into the uterus, where hopefully it'll implant and start a pregnancy. This diagram really just pulls the whole cycle together, doesn't it? You can see it all, from getting the eggs to the fertilization in the dish, through those first few days of growth, and then, finally, the transfer back into the uterus, where the next phase of the journey hopefully begins. But here's a key detail. Not all embryos make it to this final step. The embryologist really acts like a gatekeeper here. They carefully grade the embryos based on how well they're dividing, their structure, their overall quality. Only the best-looking ones, the ones with the highest chance of success, get chosen for the transfer, which is done very gently with a thin, soft catheter. Now, everything we've just gone over is pretty much the standard IVF playbook. But of course, the technology has kept evolving, and that's led to some really advanced techniques. And believe it or not, a whole range of uses that go way beyond just human infertility. One of the biggest leaps forward is a procedure called ICSI. You could think of standard IVF like putting a bunch of people in a room and hoping two of them hit it off. ICSI, on the other hand, is like a personal introduction. It's used when sperm might need a little extra help. An embryologist literally picks out a single, healthy-looking sperm and injects it directly into the egg. And the precision needed for ICSI is just, well, it's mind-blowing. On the left, a tiny pipette holds the egg perfectly still, and on the right, a microscopic needle pierces the outer shell to deliver that single sperm. It's an absolutely stunning feat of engineering on a microscopic scale. And the impact of this science is huge. These same core ideas are used for so much more. They're used to improve livestock breeding, to screen embryos for serious genetic diseases, that's called PGD, and they're even being used in conservation efforts to help save endangered species. It's an incredibly powerful and versatile scientific toolkit. But, like any really powerful technology, IVF isn't without its complications. It comes with incredible promise, for sure, but also some pretty significant challenges. So let's zoom out and take a look at the bigger picture, the promise and the perils. It really is a balancing act. On one side, you've got these incredible advantages, helping people overcome infertility, rescuing the gene pools of animals, and allowing for a powerful genetic screening. But on the other side of the scale, there are real risks: a higher chance of twins or triplets, the potential for damaging the embryo during the process, and, of course, a whole host of really complex ethical questions. And those ethical issues, they are not small. This technology forces us to ask some profound questions about the creation and often the discarding of unused embryos, and it brings up the potential for being used for things like non-medical sex selection. These are deep, tough societal debates that we are still right in the middle of. And that leaves us with a massive question for our future. IVF has helped create millions of families, and its power is only going to grow. As science keeps pushing the limits of what's possible, the challenge for all of us is to figure out where we draw the ethical lines. How do we balance this amazing medical possibility with our moral responsibility? That's the conversation we need to keep having. Thanks for joining me on this one.